And we're back. This is Dr. Jenkins. Here we go for our last chapter in the unit on the cardiovascular system. So we started with the blood chapter, then we did the heart, and now this chapter is on the blood vessels. A couple notes before we get started. In the lecture for blood vessels, we're going to talk about the structure of the blood vessels. We're going to talk about blood pressure regulation and those sorts of things. The actual names of the blood vessels themselves, that is what you're going to cover in lab, all right? So in lab, that's where you're going to learn the names of the blood vessels: celiac trunk, superior mesenteric artery, brachial vein, and so forth, right? But here in lecture, we're going to be talking about the structure of arteries, veins, capillaries, fluid exchange, and blood pressure regulation. Here's our outline for this lecture chapter. I said this before, and I want to make sure I say it again. This chapter can be sneaky hard. I think for a lot of people, you think, well, how hard can it be? There's just some blood vessels. Well, what's the structure? Blah, blah, blah. But actually, when we get into regulation of blood flow and fluid exchange, it can get pretty tricky. All right, so just be prepared. Make sure that you're spending enough time on this. And off we go. Okay, here's some fun facts. When you see things like this, they're not going to be on an exam. It's just some interesting information for you. We have a lot of blood vessels. If we were to lay them all out, it would probably be somewhere around 60,000 miles. So we have a lot. And in fact, the blood circulates through them pretty darn quickly. All right, so most of the time, blood travels through what we're going to consider a simple route. And these should be arrows, which means that blood that leaves the heart through arteries. Arteries then empty into capillaries, where fluid exchange takes place. And then the deoxygenated blood leaves the capillaries into the veins, which bring that deoxygenated blood back to the heart. So this is what we're looking at letter A. We can use green. So blood that leaves the heart through arteries. And we already know that when the blood vessels are colored in red, almost always, that means that the blood is oxygenated. And then those arteries lead to a capillary bed where oxygen is dropped off, CO2 and other waste products are picked up. Then that blood that is deoxygenated now leaves the capillaries through veins, and the veins take the blood back to the heart. So that is what happens most of the time. We're going to talk about one special instance called a portal system. We have two portal systems in our body. One of them is called the hepatic portal system. And we know by that word hepatic, we're talking about the liver. So we'll talk about that in the lab. And then a second portal system we have is between the hypothalamus and pituitary gland, which we're not going to be talking a lot about in this chapter, but we will see it rear its head again when we get to the endocrine system later this semester. So what a portal system means, it's the exact same beginning. Blood leaves the heart through arteries. That blood goes to one capillary bed. But what happens in a portal system, instead of there being just one capillary bed, there are two, right? So the blood goes through one capillary bed, and then through usually what's called a portal vessel, it goes to a second capillary bed before going into veins to return to the heart. In the case of the hepatic portal system, this arrangement allows us to filter the blood from the GI tract and the liver before it goes back into general circulation. So usually we see this when we need extra filtration. But for you, right now, just know that a portal system means that instead of just one capillary bed, there are two capillary beds. And then know our two examples, the hepatic portal system, and then there's a portal system of two capillary beds between the hypothalamus and pituitary glands. Now, just make sure that you know a definition, anastomosis. Sometimes this is defined as where two blood vessels merge, but it can also be defined as creation of new blood vessel. Because in the process of two blood vessels merging, we create a new one. But a good example of an anastomosis is if there's new tissue being generated or there's a problem with vessels that are currently there, we can simply reroute the blood by forming new blood vessels, by merging or rerouting blood flow. All right. Okay, so now let's talk about anatomy. So we're going to be talking about three types of blood vessels: arteries, veins, and capillaries. But before we get into the specifics of each of those, I want to address the layers of a blood vessel wall. And we're going to be talking mostly about arteries and veins here, okay? But arteries and veins, despite them being a little bit different, they have the same three layers in their wall. Make sure you know these three layers. What I've done is I've put in red what you need to know. So even though I'm going through it and I'm talking about what's most important, now there's really no excuse, my friends, because you can see I have put in red what's most important. So let's first get our bearings here. If we look on this picture, it's a real picture, right? And we can see that this here is an artery. We'll put that in red. And what we're looking at here, we can't see all of it, is a vein. All right? Because in the body, arteries and veins tend to be side by side, right? So the innermost layer of both of them is called the tunica interna. Interna tells us that it's the innermost. It tends to be a relatively thin layer. Here I am outlining the tunica interna, or the innermost layer of an artery. Let me just go to the next picture here. This is also a good picture of the artery versus vein. But we can also see a vein. Let me go to green. And the innermost layer would be right here. Point being, the innermost layer is thin, and it is usually made up of epithelial tissue, particularly simple squamous. So innermost layer, thin, made up of simple squamous. I know some people say squamous, I say squamous. Both mean the same thing. The next layer is the tunica media, which is the middle layer, and it tends to be the thickest. So all that I'm filling in here is the tunica media, and it has smooth muscle. We're going to see a thicker layer of smooth muscle in arteries versus veins, because arteries rely on smooth muscle to help push the blood through the vessel. But indeed, veins do have a little bit of smooth muscle also, just not as much. The outermost layer is called the tunica externa. Sometimes it gets the name adventitia. And this is the outermost layer. It is made up of connective tissue, and its job is to really connect that blood vessel wall to the other tissues of the body. So here it says anchors blood vessel to other tissues. In other words, we don't want this outer wall of the vein just to be flowing freely in the body. That tunica externa actually connects to the outer tissue. Maybe it's adipose tissue, maybe it's other connective tissue. Here's another great picture so we can see the differences between the artery and vein. Both have a thin inner layer called the tunica interna. Here's the tunica interna. Both have a layer of smooth muscle, tunica media, which is thicker in the arteries, but it's still present in the veins. And then we have the outermost layer, tunica externa or adventitia. Excellent. Now, when we compare arteries and veins, we'll talk about capillaries in a minute. Here's a nice comparison, all right? Arteries overall have thicker walls because arteries have blood going through it under a higher pressure, so it needs thicker walls in order to withstand that pressure. If arteries have thicker walls, that means that the veins have thinner walls. Arteries have a thicker tunica media. Veins have a thinner tunica media, so this should be like a down arrow, less smooth muscle. Veins do have some smooth muscle in the tunica media, it's just not as much smooth muscle as the arteries. Some other important differences. Arteries will pretty much always retain their cylindrical shape. So even though they're not as wide overall, so if I go back here, look at the artery. It's not as wide overall. Veins tend to be much wider. Look at how much wider it is. So veins tend to be wider overall. But the difference here is that the arteries, no matter if there's blood going through them or not, they will stay open. The difference here for veins, they can be wider when they're full of blood, but when there's not blood going through it, the veins will flatten. And lastly, veins contain valves to prevent backflow. Arteries do not have valves. In terms of function, this means that the arteries are much more contractile. When they fill with blood, they will get a little bit bigger, but they never collapse. They're able to kind of contract back to that resting length. It's always going to be open. They're much more resilient, thicker tunica media, and blood courses through them at a higher pressure. Now, veins have blood going through it at a lower pressure. The veins are a little bit less resilient. We could argue that they're a little bit more fragile. They are able to stretch a little bit, but they're not elastic, so they don't recoil. And a last difference I want to point out, at any given point in time, there's going to be a higher volume of blood in the veins. There is a lot of blood going through the arteries, but it just tends to go through quickly in spurts, whereas the veins, the blood travels more slowly. And this has the effect that at any given snapshot in time, there's a higher volume of blood in our veins. All right, now on the other end here, we have capillaries, which look much different. Their walls are overall very, very, very thin. And that's because we want to be able to exchange things through it. Because it is indeed in the capillaries where we unload oxygen, where we pick up carbon dioxide and other waste products. That could only happen through a thin blood vessel wall. The other difference between capillaries and the others is that they form beds, right? So instead of just being one straight vessel, in capillaries, they form a network where there's a lot of smaller vessels, which of course increases surface area, means we can do a lot of exchange. Excellent. Now, there are different types of arteries, elastic, muscular, and arterioles. Although they have the same structure, they all have the same layers. Again, I apologize for that message that keeps coming up. Please ignore. All three types of arteries have the same structure: tunica interna, tunica media, tunica externa, thick, smooth muscle, keep their shape, strong, have blood moving under high pressure. But it really has to do with their size. And here's how I remember these. I just look at the first letter of each: elastic, E, muscular, M, and arterioles, A. And that gives me Emma. Maybe this works for you. It's always helped me to remember. The arteries that are closest to the heart are considered to be elastic. They are the biggest. Because they're closest to the heart, they're simply going to be transporting more blood. As we move a little further away from the heart, we get to muscular arteries. And then the smallest arteries right before they empty into the capillaries are called arterioles. Be able to give an example of each. The elastic are the biggest. They are the most elastic. They have to be because they have the most blood going through them and they're closest to the heart. They have a lot of pressure. An example of an elastic artery is the aorta, which we know leaves right from the heart. A little further distally from the heart, we have muscular arteries. These are medium in size. An example would be the femoral artery, or you could say brachial artery. So just think of major arm and leg vessels. And then the arterioles are the smallest, and it's important to note that they are right at the entrance to the capillaries. The name tells us arterioles that they're very small. And the most important thing about these arterioles is that they have the ability to constrict and dilate. And because they're blood vessels, by the way, the prefix for blood vessels is vaso. So when we say vasoconstrict or vasodilate, we're just simply talking about the fact that a blood vessel constricts or dilates. I apologize again. Apparently my cold is still here. Okay, this is really important. As we're going to find out later, it is the ability of these arterioles to constrict or dilate that controls movement of blood into the capillaries. In this way, it is wonderfully simple. Wonderfully simple. If I want blood to go into a capillary, then I'm going to dilate the arteriole leading into it, make it bigger. If there's a moment when I don't want blood to go into the capillary, then I constrict the arterioles, which means that blood cannot get through. So at all times, our body is making decisions about which arterioles we need to dilate versus which ones we can constrict, because we're always distributing blood to where it's needed the most. Now, I'm not saying that we completely cut off blood flow to parts of our body at times for long periods of time. No. We're always going to have some blood flow going into your GI tract. You're always going to have some blood flow going into the muscles of your legs. But we can change how much blood flow. Think about when you're exercising. If I'm exercising, I've got to find a way to deliver more blood to my working muscles. They're going to be working, they need more oxygen, they need more glucose. So the arterioles leading into those skeletal muscles will dilate. But because I only have a certain volume of blood, 4 to 6 liters, I have to take it away from somewhere else. So a good example is exercise. If I'm exercising, I'm going to be sending more blood to the muscles that are working, which means that I'm going to take a little bit of blood away from the GI tract. It's not as important when I'm exercising to digest my breakfast. And this is possible because of the arterioles. I mean, how cool is that? See? School is cool, I tell you. All right. This is not going to be on the test, but you can see a lot of times we get plaque buildup. That plaque buildup will begin along the tunica interna. Here's that plaque building up. Could be from a high cholesterol, high fat diet, inactivity, partially could be genetic. And of course, this narrows the opening that blood can go through. So it means not as much blood can go through. So we have a risk of limited blood flow to our body. It also means the blood goes through at higher pressure, which can be dangerous. And in advanced stages, this plaque will kind of anchor itself into the tunica media. And that means that we no longer have nice, pliable muscle. This gets hardened. And then that part of the artery overall is no longer able to contract as it needs to. Atherosclerosis is when we have this clogging of the artery. Okay. All right. Just like arteries had names, so remember the arteries closest to the heart were considered to be elastic, then muscular, and then arterioles, right at the entrance to the capillaries. Well, veins have names too. I'm not going to have you know these because it's not as important in terms of blood flow regulation. We can simply just say that the veins closest to the capillaries are smallest, and then they get bigger and bigger as we go towards the heart because they're picking up more and more blood. So you don't need to know the exact names, but in a similar way, the veins start off one size. In this case, they start off small and they get bigger. Now, the capillaries. We've already talked about them. We said that they have very, very thin walls. They form beds. So we can see here they form this bed, so to speak, which increases surface area. We have so many capillaries. And because we have so many capillaries, it's almost like in our body, these capillary beds tend to be pretty close to each other. And that means that any little cell has reasonably close access to any capillary, which is a good thing. There is something called a precapillary sphincter. A sphincter is a thickening of muscle. Let me see if I have a picture on this next one. Yeah, here we go. So a sphincter, it looks like these little bubble things. It almost reminds me of a Kong dog toy, right? But a precapillary sphincter is just a thickening of muscle. It's another way of regulating blood flow. It's just that instead of, for example, in the arteriole, the entire arterial outer wall can constrict or dilate. That's one way to regulate blood flow. Well, right at the entrance to the capillary, we have this thickening of muscle around the capillary wall, and it can contract or relax. Because similarly, we need to be able to control when and how much blood goes into each capillary bed. All right. There are technically different types of capillaries. We do not have to know this now. We are going to come back to this because when we get to the kidneys, for example, we're going to talk about fenestrations. Or, hold on for 1 second. Excuse me. Or when we talk about the liver, there is a specialized capillary structure called a sinusoid. So we don't need to know this yet, but what we're going to find later is that some capillaries have a different structure, which makes the pores in the capillary walls bigger or smaller. So for example, in the kidneys and the liver, the pores in the capillary walls are pretty darn big, and that just helps filtration. Because after all, what is the job of the kidney and the liver? Filtration. Whereas most other capillaries around the body have smaller pores in their walls. But you don't need to know this for now. Just a little poke so that when I come back to it later this semester, hopefully you at least have some remembrance. Oh yeah, she did say there were different types of capillaries. All right. I alluded to this earlier. Excuse me. It's funny, it seems like my nose just starts to run when I'm about to start talking and give this lecture. So I alluded to this earlier, and now I just want to show you it in a visual way. If we were to look at the blood distribution in the blood vessels at any given point in time, most of our blood is found in our veins. And that's just because they open up, they're very wide, And because the blood moves more slowly through veins, we just happen to have more in there at a given time. So I'm not going to ask you this exact percentage per se, but have an idea. I mean, this is really like 3/4 of all of our blood, just about. And you can see that that's shown in the blue here. So all of this blood is in the veins, and we have much less in the arteries, pulmonary vessels, and capillaries. Okay. Now I want to begin this discussion, and I will have a second blood vessel lecture, but I definitely want to start this because this is where it gets a little tricky. The structure of the blood vessels is straightforward, but here we're going to get a little tricky. All right. There's a few terms I want to start with. When we talk about blood flow, we're talking about the amount of blood. So we could say an area of the body is receiving a high blood flow. And just substitute the word amount. So if an area of the body is receiving high blood flow, it's receiving a large amount of blood. Perfusion is a little bit different. Perfusion means that we're also exchanging. So we're infusing. So it talks about rate of exchange at capillaries. So there's two important parts to this definition. Rate, it's not necessarily the amount, but it's the rate. And it's what's the rate of exchange at the capillaries. When we talk about these principles, sometimes these will go hand in hand. Obviously, if there's a larger blood flow to an area, we're able to do more perfusion. But sometimes it doesn't go hand in hand. Sometimes you can have an organ with a relatively low blood flow, but even though there's a relatively low blood flow, whatever blood flow is getting there is perfused at a high rate. Okay, here are some things, but I'm not as worried about that right now as just the definitions up top. Here are some examples. If we look at the femur, for example, the femur gets a lot of blood flow, and the blood flow that gets there is perfused quickly. There's a lot of exchange into the capillaries. The ovaries, well, in a female, the ovary is a pretty small organ. So relatively, it's got a low blood flow. But with that blood flow that it gets, it has a high perfusion. So it's almost like the perfusion tries to balance out the fact that there's less of a flow. Skeletal muscles get a high flow and a high perfusion. So, this is a good example of where there would be a little bit of a discord. And it's simply just because the organ is pretty small. The ovary is relatively small as an organ, but it's very metabolically active. It needs a lot of oxygen and nutrients. So even though it's not a whole lot of blood flow going to it, there's a high perfusion rate. All right. In order to talk about the principles of blood flow, we need to introduce some principles of fluid dynamics. Now, we did a little bit of this in the heart chapter, whether you liked it or not. We talked about pressure and volume, remember? So when we talked about the cardiac cycle, we said that we can alter the volume of a chamber, which therefore alters the pressure. Because when volume is high, pressure is low. When volume is low, pressure is high. And then we can develop a pressure gradient whereby blood moves from high to low pressure. Well, what we're going to find is blood in the blood vessels also moves from high to low pressure. but there's a little bit more that we need to talk about. So I'm going to first introduce this, I would say equation, but whatever you want to call it. And unfortunately, the symbols didn't show up when I brought it over to my iPad here, but we can draw it out. Flow is proportionate to change in pressure over resistance. This is what it's trying to say there on the left, but the boxes overtook my symbols. So proportionate to is that little infinity symbol looking thing. Blood flow, fluid flow, is proportionate to change in pressure over resistance. We could pretty much treat this as if the proportionate to was equals. So it tells us that flow and change in pressure have a direct relationship. So when blood flow is high, we tend to have a higher change in pressure or pressure gradient. And I'm going to give you some examples later on, but if we can just learn these principles now, it'll make our discussion later a little bit easier. When there's a high flow, there's a high change in pressure. Because there's a direct relationship, we could just alter this and we could say when there's a low flow, there's a low pressure gradient. So they're directly related. On the other hand, flow and resistance are inversely related. So that means that, sorry, I had some cat hair on my screen. This means that when there's low flow, there is high resistance. Or the alternate is true. When there's a higher flow, there's less resistance. Okay? So I've put them all together here, right? Flow is high. We have the highest blood flow when there's a high change in pressure and low resistance. We have a slower blood flow when there's a lower change in pressure and higher resistance. You can see here I have change in pressure as blood pressure. That's another way of thinking about it. And I think this can make sense if we have the heart, and we do have a heart, by the way, hopefully, and then we have the aorta, which is the big artery coming out of the heart, right? So how is blood going to flow? Blood's going to flow in this direction. The blood moving through the aorta will have a highest flow, meaning we can most easily get the blood through the aorta when there's a high change in pressure and low resistance. Well, of course there's a high change in pressure. We're right by the heart. So we have the highest pressure out of any blood vessel right out of the heart. And anytime we get further away from the heart, the pressure goes down a little bit just because of the fact that we're moving away from something. So we do have a pressure gradient. One side it's higher, other side it's lower. And there should be low resistance. We don't have any atherosclerosis. There's not high systemic blood pressure. So this blood leaving the aorta, traveling in the aorta from the heart, is going to be at the highest flow when there's a big change in pressure and no resistance or little resistance. Well, what are some things that could make the blood flow less? What are some things that would make the ability of blood to leave the aorta less? Well, we would have less blood flow. Instead of having 3 arrows of blood flow coming to, what if I only have 1? We would have less blood flow if there is resistance or some kind of pressure coming back. We might add resistance by something that causes afterload. think back to our heart chapter. Afterload is any resistance that the blood encounters on its way out of the heart. Maybe there's increased resistance because of plaque in the arteries. Maybe there's increased resistance because of overall high blood pressure in the body. Well, if we have resistance, that means that the blood coming out of the heart has to work harder to get the same blood flow through that resistance. All right. It's going to get a little tricky because as you're going to see, we're going to add some more equations to try and help us understand this. One way that I think people can help themselves, and I know when I was a student, and by the way, I got my PhD a couple years ago, so I went back to school 14 years after my master's degree for my PhD. So as many of you know, going back to school after a 14-year break is a little challenging. So it was a bit of a learning adjustment, and it's definitely made me a little bit more empathetic to you students. So I'm not that far away from studying. What has helped me, and it may help you, is, for example, when you get to a test, on the side of the test, if we're in person or on a blank sheet of paper, because you're not going to cheat, just write down these. So on the test or on a spare sheet of paper, before I even start my exam, I might actually write down this. I may write that down. And I may even write down, okay, so we have a high flow when there's a high change in pressure but low resistance. And in writing it down, first of all, you unload it. So if you're you might be stressing trying to remember it. But if I write it down, well, then it's unloaded from my brain. So there's a little bit of relief there. But then half an hour later, when I get to a question on the exam about this, I'm visual. I may actually go back to look at this. And the question's talking about when is flow high? Well, then I can look at my notes. Oh, right, flow is high when the change in pressure is high, but resistance is low. So it might be helpful for you to write this down. Another thing that's been helpful is, and you may be wondering why I have a picture of a garden hose, the fluid dynamics that we're discussing are very similar to the fluid dynamics of water coming out of the garden hose. So if I want to talk about flow of water through a garden hose, the flow of water through a garden hose is highest when there's a high change in pressure. And I can create a high change in pressure by turning the spigot on as high as it goes. So I bring in as much water as possible. So there's a really high pressure right at the spout, and then the pressure gets lower the longer we go along the hose. Okay, so flow through a garden hose is highest when there's a high change in pressure, when I really crank open the spigot and have a high pressure of water coming through. The more I open up the spigot, the more flow comes out, right? If I turn the spigot down a little bit, there's not as much water coming out, so there's less of a pressure gradient, so there's less flow. However, what if I consider flow and resistance? What is one way in a garden hose to add resistance? If you squeeze a part of the hose. If you want to spray someone with water, you squeeze the end of the hose and the water comes out faster. Well, that's a way of adding resistance. If the opening of the garden hose is like this, but then I squeeze it, I'm decreasing the opening of the hose. So that's a way of adding resistance. When I add resistance, when I squeeze the end of that hose, what happens to the amount of water flow coming out? If I squeeze the end of the hose, not as much water comes out. The flow goes down. So when I add resistance, when I squeeze the end of the hose, there's less flow of water coming out. Now, the water that does come out comes out with a higher pressure. That's true. but I'm talking about flow. When I squeeze the end of the hose, less water comes out. This is a tough one, okay? Spend some time on it. It's only easier for me because I've been teaching this for years. There's a review handout on Blackboard to help you practice these principles. This is just saying the same thing as I wrote down here, okay? So no need for me to go over that again. Now, we're also going to be talking about blood pressure. So just ignore that, please. We just talked about blood flow. So blood flow is one consideration. Flow is the amount of blood. And as we know, flow was related to or proportionate to change in pressure over resistance. Well, we're also going to be talking about pressure in the blood vessels, because both of these things matter. It's not just the flow that we care about, or how much blood is going through at a given time. It is the pressure with which that blood is going through. So we're going to give another equation for pressure in a minute, but first, let's just define it. When we talk about blood pressure along a vessel, we're talking about the force that blood exerts onto the vessel wall. I think you understand this. A high blood pressure just means that the blood going through is exerting more force against the arterial wall. We have much more pressure. There's much more force of blood against the vessel wall and arteries, and there's much less pressure in veins. When we do a blood pressure reading, we get a number that has a numerator and a denominator. The normal is around 120/80, and it's measured in millimeters of mercury. That top number is referencing the pressure in the arteries during systole. Let me get my phrases straight. So the top number is the blood pressure in an artery during ventricular systole. So wouldn't there be a higher arterial pressure right after the heart's contracting? Of course. And the bottom number is referring to the blood pressure in an artery during ventricular diastole. So know that. Know what a normal blood pressure is, a roundabout. We can also measure something called mean arterial pressure, but you don't need to know this. Now, as we get further away from the heart, the blood pressure goes down. I think this makes sense, everyone, right? I mean, if we look at the heart, we have the aorta leading right away from the heart. Well, of course, the pressure in the aorta is the highest because it's closest to the pump. But look at how drastically the blood pressure goes down, right? Anytime we have a pump and it pushes something, well, the pressure is going to be highest closest to the pump. And just through the nature of resistance and physics, as we move something through a tube further away from the pump, we increase the resistance and the blood pressure goes down. So it's very significant. Very little pressure in the veins. Very high pressure right outside of the heart. I'm not going to ask you any of this specific chart, but it does prove a point, right? That the blood pressure goes down very quickly and that there's very low pressure in the veins. Speaking of systemic blood pressure, make sure you know these conditions. You may already. Hypertension is a chronic resting blood pressure that's too high, and hypotension is a chronic resting blood pressure that's too low. Notice we're talking about a resting blood pressure. When we exercise, we want the blood pressure to go up, but we're talking about just when someone is sitting down, not doing really anything. High blood pressure just sitting is very bad because it puts a lot of pressure on the blood vessels, which means the heart's got to work harder, and over time it can lead to a lot of negative consequences. Here we go, my friends. So we talked about, and I'm going to write this down again, we talked about a principle of blood flow. And we said that flow is proportionate to change in pressure over resistance. Flow is one principle. Well, now we're adding a second principle, and that is one of pressure in the blood vessels. Blood pressure is equal to our cardiac output, which we talked about last chapter. We might also see it as a Q with a line over it, times peripheral resistance. Right? So this is where I'm suggesting that you write these two down from memory. So that way when I ask you a question, you know which equation to go to and you can make better sense of it. So if we just look at this, we can see that both cardiac output and peripheral resistance have a direct relationship with blood pressure. So if I increase my cardiac output, if I increase the amount of blood being pumped out of the heart, well, it's going to increase the blood pressure. If I pump more blood out, there's going to be more pressure in the vessels. Think about during exercise. Your heart's going to pump out more blood more often, and therefore the pressure of the blood in the vessels goes up. Likewise, resistance. Peripheral resistance is just referring to resistance overall in the periphery, but if it makes your life easier, you can just think of it as resistance. When I increase resistance, I also increase blood pressure. Let's think about the garden hose. Same principles here relate to the garden hose. If I open up the spigot, that means I'm increasing cardiac output, right? If I increase the flow of water coming out, I'm going to increase the pressure of the blood in the vessels. If I turn the spigot down and I have less blood flow or less water flow, then there's less pressure. You know this, when you turn the spigot up and there's more water coming out, you can feel the increased pressure of the water inside. Whereas if I lower the spigot and I have less water coming out, then there's less pressure. Resistance, I gave you the example before of squeezing the end of the hose if you want to spray somebody with water. Well, when I add resistance, it increases the pressure. So when I squeeze the end of that hose, there's less water coming out, so there's less flow, but whatever water is coming out comes out at a higher pressure. Everyone knows that. When you squeeze the end of a hose, the water coming out comes out at higher pressure. That's why you do it. So you want to look at both of these, right? Because they can sometimes get confusing. It particularly gets confusing when we talk about resistance, right? Because, as we talked about here, when I increase the resistance, I increase the pressure. But what about flow? When I increased resistance, what does it do to blood flow? It goes down. So with resistance, it does one thing to blood pressure, it does the other to flow. And that's where I really think the garden hose helps. If I increase resistance, if I squeeze the end of the garden hose, if I increase resistance, there's less water coming out, but whatever water is coming out comes out at a higher pressure. So anytime you add resistance, it reduces the amount of blood. It reduces the flow, but it increases the pressure. Again, please refer to the handout on Blackboard. All right, we're going to finish a couple of things here, then we're going to end the first video, and then we'll pick up the second video with blood pressure regulation. When we talk about this equation, blood pressure can be influenced by a lot of things. We can influence blood pressure by changing how much blood we pump out of the heart. We can alter blood pressure by changing the resistance that blood encounters. I'm going to point out a couple things here. Viscosity. We talked about this in the first chapter. Thickness. Anytime the viscosity of the blood goes up, which is another way of saying the blood is thicker, that's a way of increasing resistance. Think about it. Trying to pump water through a hose is easier than trying to pump maple syrup through a hose. Because maple syrup is thicker, it's a way of adding more resistance, and it's harder to get that flow through. So if I increase viscosity, it increases the resistance. And what does that do to pressure and flow? Well, increased resistance will decrease flow, so not as much maple syrup comes out, but the pressure goes up. So when we have changes in viscosity, like dehydration, because there's less water content in the blood is thicker because there's less water to balance out all the formed elements. So if we have thicker blood during dehydration, that's a form of resistance. It increases the blood pressure, which can have negative consequences, and it means that less blood is going to be able to pass through, which can also have negative consequences. But far and wide, the biggest thing that influences pressure and resistance is the vessel radius. You can see I skipped vessel length. That means you don't need to worry about it. The biggest thing that influences pressure and resistance is the vessel radius. So this is what influences resistance and pressure the most. And we make use of this, right? We talked about the arterioles, if you remember, and of course you do. The arterioles are the smallest arteries right as they enter the capillaries. And the arterioles have the ability to vasoconstrict and vasodilate. And this is a way that we can regulate pressure and flow. So what's happening when an arteriole vasoconstricts? Because like I said, this is a main way. This is a major way of how we regulate blood flow and pressure. If I need to bring in more blood flow to an area, I'm going to dilate the arteriole. If I need to bring in less blood flow to an area, I'm going to constrict an arteriole. But we can also vasoconstrict and vasodilate these arterioles as a way to regulate blood pressure. And you may not have thought of that, but we definitely do it. So let's get these clear before we do the next video on blood pressure regulation. When an arteriole vasoconstricts, we are increasing resistance. Think about a normal diameter of an arteriole. Well, if I vasoconstrict, it's going to be smaller. So I'm asking the same amount of blood to go through a smaller tube, increases the resistance, which means that the flow goes down. If I narrow that vessel, less blood is getting through, but the pressure going through that tube goes up. Think of the garden hose. If I squeeze the end, if I add resistance by squeezing the end, there's less blood flow, less water flow, but there's more pressure coming through it. On the other hand, dilation, if I dilate, I make it even bigger, right? This was normal or resting. So if the diameter now becomes bigger, I've lowered the resistance, which means I've increased the flow. If I have a bigger tube, can I fit more blood through it? And because wait a minute, let's make sure I get this right. Vasodilation lowers the resistance, increases flow, lowers pressure. See, even I had to think back to those equations for a second. So you're not alone. So if I dilate the vessel, there's more blood flow coming through, and the blood flow going through is at a lower pressure. So we're incorporating both of these. These principles, ladies and gentlemen, is how we regulate flow and pressure. And this is vital. The blood flow, it's our lifeline. It carries oxygen, nutrients, everything. So our body has to be able to send the blood to where it's needed at the right time and in the right amount. And that is not an easy job, right? Think about all of our blood vessels like pipes, and our heart is like the spigot. So we have a certain amount of blood coming out of our heart every minute, but then our body's got to make the decision of where it needs to go and how much based on thousands of situations. And this is how we do it, by regulating pressure and flow largely through vasoconstriction and vasodilation. All right, we're going to end there for the first video, and we'll come back, and then we're going to talk about blood pressure regulation. I can't wait.